Hello everyone, I am Dr. Karthika from Department of Biomedical Engineering, SNS College of Technology, Coimbatore. In this May 2026 lecture series, we are going to discuss about glucose electrodes in bioelectric measurement and biosensor in our course Biomedical Transducers and Sensors. See, our real world problem is diabetes affects 463 million people worldwide, which requires continuous blood glucose monitoring and critical for preventing life threatening complications. And who is affected? Diabetes patients and clinicians. C. The limitations of existing methods. The limitations of existing methods, which includes lab test, fasting glucose, and urine glucose. And lab test involves delayed results, inconvenient, and high cost. Fasting glucose, which includes single measurement, no trends, incomplete data, and urine glucose, which is not accurate, lagging indicated, and hours of delay. Our clinical challenges a point of care testing, minimally invasive, continuous monitoring, and real-time data. Here, see the table, traditional versus point of care. Here I've included the parameters, a result time, location, convenience, cost, as well as trend analysis. The traditional, which has hours to days, location, the test should be carried out at the laboratory. Convenience is low, cost is high, and trend is no. And point of care, which is measured in seconds, and we can analyze at our home or bedside. High convenience, low cost, and trend analysis is. Here, see the glucose monitoring workflow. First one is the blood sample and enzymatic, which is processed in electrochemical reaction, and finally we'll have a display output. See the definition: electrochemical biosensor that converts glucose concentration into measurable electrical signal using enzyme based transduction. Our main thing is to convert our electrochemical biosensed output from our measurable electrical energy. That's it. This is the main basic principle of our sensors and transducers. Biosensors gets the electrochemical reaction, then which sends the information. Here the sensed information is converted into electrical signal with the help of transducers. See the diagram of glucose electrode structure and working principle. First step, enzyme catalyzes glucose oxidation. Step 2, it produces H2O2 plus electrons. Step 3, current and glucose concentration. Current is directly proportional to the glucose concentration present in our sample. The characteristics, which includes high specificity, fast response, portable, and low cost. Key components involves the working electrode. It might be platinum or gold, a reference electrode Ag or AgCl, enzyme layer, a glucose oxidase, and membrane is selective and protective. C. The glucose oxidase enzyme, specific to beta D glucose. It uses FAD coenzyme. C. Here the FAD coenzyme oxidizes to gluconic acid and finally regenerated in cycle. See the reaction mechanism: glucose plus O2, gluconic acid plus H2O2. Enzyme substrate complex, gluconic acid is electron transfer, and finally H2O2 production. Here the immobilization techniques includes physical absorption, covalent binding, and entrapment and cross-linking. The advantages of enzyme electrodes includes high selectivity, fast response, mild conditions, and reusable enzyme. See the working electrode materials. We may include platinum, excellent conductivity, inert. Gold is biocompatible, expensive. Carbon is low cost, wide range. Silver is a reference electrode. Enzyme layer, which has glucose oxidase enzyme, immobilization matrix, protective membrane, and enzyme stabilizers. See the diagram of test strip cross section. Here the substrate materials used are plastic, flexible, low cost, ceramic, robust but high temperature. See the material selection criteria. We have to consider the bioconductivity, biocompatibility and stability. See the working principle. See here. See the signal flow. First, our thing is our blood sample, which is fed into enzyme layer, then electrochemical cell, which is amplified, signal is processed, finally it will get displayed. See the measurement step. Blood sample, enzymatic H2O2 production, electron transfer and current, current reduction amplify, signal conversion to value, and then digital concentration display for measurement. See the types of glucose electrodes. See the types. See the types. We have I've listed a table. First type is amperometric. Second one is potentiometric. Third one is optical. Here these types are differentiated based on sensitivity, cost and applications. See the applications in healthcare, self-monitoring blood glucose, continuous glucose monitoring, hospital monitoring, screening as well as diagnosis. Here I have included student activity, group discussion for 5 minutes. See the assessment questions, gate style, industry as well as conceptual based on glucose biosensors. See the references and summary part. These are all the references and summary. Summary part we have for principle, advantages, challenges, types and applications. The main thing is which is used in continuous glucose monitoring, hospitals as well as screening. References for our glucose electrode. Thank you all.